After studying this module, you shall be able to learn chemical control of wakefulness and arousal, learn chemical control of REM sleep, learn various brain structures and neurotransmitters that control desynchrony and muscular paralysis in REM sleep, learn about the PGO waves initiating REM sleep, learn about the neurochemistry of non-REM sleep, particularly slow wave sleep, learn about the effects of various drugs or chemicals that affect sleep. Sleep is a natural, periodically recurring state of inactivity characterized by the loss of consciousness and reduced responsiveness to external stimuli. In contrast, wakefulness is the absence of sleep and is marked by consciousness, awareness and activity. Regulation of sleep-waking cycle is complex and involves diverse brain circuits and molecules. Many neurochemical systems interact to generate wakefulness and sleep. This complexity involves an interplay among many neuroanatomical and neurochemical systems. The total duration of sleep is regulated in terms of time. One attempts to make up for the deficit by sleeping for longer hours. A physiological base in terms of some chemicals is essential to maintain this balance. Studies to find out such substances have now been focused on the chemicals of the brain. Some earlier investigations revealed that the two hemispheres of the brain may go to sleep by turns. Had it been some substance in blood, it would have reached both the hemispheres via a general system of circulation. Studies have further indicated that accumulation of a sleep-producing substance during wakefulness, which produces sleep and then gets gradually destroyed during sleep. When it has been fully utilized, the individual gets up. Another chemical substance called the wakefulness producing substance, which gets accumulated during wakefulness and keeps getting diminished till it finishes and produces sleep, has also been discovered. Specific regions of the brain for the production of such substances, even specific to the two hemispheres, have been identified. Chemical control of wakefulness. Waking and consciousness depend on the activity of neurons in the ascending reticular activating system of the brain stem. These neurons project into the thalamus, hypothalamus and basal forebrain and eventually send projections to the cortex. There are particular neurotransmitters such as catecholamines, acetylcholine, histamine, glutamate and aspirate that are localized with the reticular formation and have important roles in cortical activation and arousal. The neurotransmitter acetylcholine plays an important role in this context. The groups of neurons on pons and basal forebrain produce cortical activation desynchrony when they get stimulated. Hippocampus, which is involved in arousal, learning and memory processes, receives input from neurons in the medial septum. Day, Damsma and Fibiger studied the areas involved with alertness, which comprised a striatum, hippocampus and frontal cortex with the help of microdialysis. They highlighted the role of acetylcholine in maintaining and producing levels of alertness. Rasmussen, Klo and Spurg found that the level of acetylcholine increased by 350% when cerebral cortex was activated by neurons coming from the dorsal pons and which contained acetylcholine. The importance of adrenergic neurons has also been highlighted by researchers. A major center on dorsal pons, the locus coeruleus, is discovered to be strongly related with the levels of arousal. It projects to neocortex, hippocampus, thalamus, pons, medulla and cerebellum via neurons that are primarily adrenergic. The main neurotransmitter involved is thus norepinephrine. When a sleeping animal is suddenly awakened, there is a spurt in the level of norepinephrine in these neurons. The activation of these neurons was highest when the animals were in vigilant state rather than when they were engaged in an ongoing activity. The effect of awakening an animal from the REM sleep and related increase in norepinephrine has been shown in many investigations. Besides acetylcholine and norepinephrine, another neurotransmitter involved with vigilance is serotonin. The seat of serotonin producing neurons is raphe nuclei located on pons. These nuclei project to several areas of the brain including thalamus, hypothalamus, basal ganglia, hippocampus and neocortex. These neurons maintain the ongoing activities during wakefulness such as eating food. However, when something happens suddenly and vigilance is immediately required for the purpose of survival, such as another animal trying to snatch away the food, neurons containing norepinephrine located on the locus coeruleus of the pons become at once active. The neurotransmitter histamine plays a crucial role in the process of arousal. The neurons containing histamine are located on the tuberomammillary area of hypothalamus rostral to mammillary bodies. This area is associated with cortical arousal. Neurons in this region also project to various areas of the cerebral cortex. Most of the drugs to treat allergies are antihistamines and therefore reduce arousal and produce sleepiness. Thus, acetylcholine, norepinephrine, serotonin and histamine containing neurons located on pons and hypothalamic areas projecting to higher centers constitute the basis of arousal systems in the brain. The actions of some familiar drugs emphasize the neurochemistry of arousal and wakefulness. For instance, drugs such as cocaine and amphetamines increase levels of catecholamines. These drugs are important in pathways involved in behavioral arousal and cortical activation. These drugs decrease total sleep time, increase sleep latency and decrease REM sleep. Amphetamines have therefore been used to treat excessive sleepiness seen in narcoleptics. Nicotine is another commonly used drug which increases arousal and vigilance. It acts on cholinergic receptors found in the reticular formation and the basal forebrain. These neurons are an important component of the ascending reticular activating system and are thus involved in maintaining cortical activation and wakefulness. Other excitatory neurotransmitters such as glutamate and aspartate are involved in wakefulness. Chemical control of REM sleep. The evidence suggests that nuclei located on locus coeruleus containing norepinephrine and those on raphe nuclei containing serotonin on pons which generally maintain arousal are inversely related with the REM sleep. Reduction in the rate of firing of these neurons triggers a bout of the REM sleep. Electrical stimulation of pons in the area ventral to locus coeruleus induces REM sleep. Since the main neurotransmitter here is acetylcholine, the pharmacological stimulation by a cholinergic agonist also produces prolonged REM. Studies also indicate that appearance of PGO waves, activation of neurons on pons, lateral geniculate nucleus of thalamus and occipital cortex appear only when the serotonin containing neurons are silent. The PGO waves are the first indication of the initiation of REM sleep. The neurotransmitter acetylcholine is equally important in the context of REM sleep. Acetylcholine is located within neurons in the pontine tegmentum and is involved with REM sleep generation. REM-on cells are cholinergic cells in the lateral pontine and medial medullary reticular areas that innervate the thalamus, hippocampus and hypothalamus. These cells discharge at high rates during REM and show little or no activity during non-REM phase. Histamine, which inhibits catabolic enzymes, precipitates the appearance of REM sleep during non-REM sleep. The injection of carbachol, a muscarinic agonist, into the pontine tegmentum induces REM sleep. Blocking muscarinic receptors will retard the appearance of REM sleep. The importance of acetylcholine was investigated by Sitaram Moore and Gillen in 1978. They found that an acetylcholine agonist, erolin, shortened the interval between periods of REM bouts. High levels of acetylcholine during wakefulness as well as REM sleep have been reported in several investigations. REM-off cells are noradrenergic and serotonergic cells found in the locus coeruleus and raphe nuclei. These are cells which are slow or silent during REM sleep. Affecting levels of norepinephrine or serotonin can have an effect on REM sleep. In general, antidepressants have the effect of decreasing REM sleep, which is elevated in human endogenous depression. Generally, the effectiveness of antidepressant drugs is proportional to the degree to which the drugs diminish REM sleep. Tricyclic antidepressants such as amitriptyline and imipramine block the reuptake of norepinephrine and serotonin, as well as having an anticholinergic effect. These drugs vary in their effectiveness as sedatives. The new general antidepressants, that is fluoxetine, sertraline, act specifically to block serotonin reuptake. Most of these drugs diminish REM sleep. The peribrachial area has been recognized as a significant center in sleep, particularly REM. It is located in the dorsolateral areas of pons. It is densely packed with the cholinergic neurons. These neurons, even if taken one at a time, fire at a high rate during REM sleep as well as in wakefulness. They are considered as the trigger which initiates a bout of REM sleep. Lesioning out these neurons drastically reduces the REM sleep. The region ventral to the locus coeruleus on pons, referred to as the MPRF, medial pontine reticular formation, stimulates acetylcholine neurons of the prebrachial area. Microdialysis studies have indicated increased levels of acetylcholine during REM sleep, which gets abolished if these neurons are damaged. Desynchrony of EEG waves during REM sleep. Thus, cholinergic neurons of pons consist of reticular activating system. They project to those regions of thalamus which are involved in cortical arousal. From here, the neurons project to mesopontine reticular formation containing the neurotransmitter glutamate. Thereafter, the projection goes to the basal forebrain structures which are cholinergic. Activation of these neurons in the forebrain then produces arousal and desynchrony, which is the typical feature of REM sleep. The PGO waves initiating REM sleep. The PGO waves are controlled by the projections from peribrachial area to the lateral geniculate nucleus of thalamus. The rapid eye movements are accomplished due to projection from the peribrachial area to the tectum. The neurons that mediate such projections are cholinergic. The muscular paralysis accompanying REM sleep. The muscular paralysis during REM sleep is mediated by the neurons located ventral to the locus coeruleus. Their axons travel down to the magnocellular nucleus of medulla. From here, the projection goes to the spinal cord where they form synaptic connections with the motor neurons. The muscular paralysis takes place since the synaptic transmission involves the inhibitory neurotransmitter glycine. The studies have revealed that lesions of the magnocellular nucleus produce REM sleep without muscular paralysis. The neurochemistry of non-REM sleep. The anterior hypothalamus and adjacent forebrain areas are important in producing non-REM sleep, as are the thalamus, dorsal raphe and nucleus tractus solitaries. Neurotransmitters such as serotonin and GABA, which may be important in sleep mechanisms, are located in these brain regions and play an important role in sleep. Serotonin 5-HT found in raphe neurons of the brainstem may be involved in sleep onset. Insomnia occurs when serotonergic cells of the dorsal raphe are lesioned. MAO inhibitors specific for 5-HT enhance sleep. Also, there is evidence that substances in the biosynthetic pathway of serotonin such as tryptophan and vitamin B6 may facilitate sleep. However, in spite of all the evidence supporting the role of serotonin in sleep onset, there are studies which suggest that the role of serotonin in sleep is not as clear as it may seem. The exact role of serotonin in sleep has not yet been elucidated. The inhibitory neurotransmitter GABA is released in highest concentrations during non-REM sleep. GABAergic neurons are located throughout the brain, including the basal forebrain, hypothalamus, thalamus, brainstem and cortex. Hypnotics such as benzodiazepines, diazepam or triazolam and barbiturates, phenobarbital, secobarbital tend to work by potentiating GABA-mediated inhibitory processes. Both bind to sites on GABA receptor complex, increase C1 flow through ionophore, causing hyperpolarization and reduce cell firing. They may shut off neurons in the reticular activating system and inhibit transmission and activity of neurons that project to the cortex and thalamus. Overall, hypnotics do increase total sleep time, decrease sleep latency and decrease the number of awakenings. They also decrease the amount of time spent in non-REM sleep stages 3, 4 and in some cases REM sleep, which are sleep stages necessary for normal restorative night of sleep. Tolerance to these drugs develops fairly quickly within 2 weeks, and insomnia and REM rebound are seen upon withdrawal from these drugs. Chemical control of slow wave sleep. The 4 brain structures have been shown to control the slow wave sleep. Electrical stimulation of the forebrain, specifically the ventral frontal lobe and anterior hypothalamus. Neurons in this region become active at sleep onset and release GABA, which is gamma aminobutyric acid, to suppress neural activity in the nearby tuberomammillary nucleus in the posterior hypothalamus. Many drugs used to anesthetize the patient during surgery also work by making GABA neurons more sensitive in this area. It produces a slow wave sleep like sleep state. If left at this stage, the brain will sleep forever. It is the brainstem reticular formation that arouses the brain from this slumber. Effects of drugs or chemicals on sleep. Since ancient times, people have tried to find out substances to enhance the quality of sleep. Ancient Greeks used several parts of the poppy plant for the purpose. The products of the mandrake plant, today known as scopolamine and atropine, have also been used to promote sleep. Some drugs developed to cure anxiety reactions were found to be having the hypnotic effects also. The drugs such as Valium, Librium, Halcyon, Ambien Sonata and Lunesta belong to the category of benzodiazepines. Such drugs increase drowsiness. They affect other aspects of sleep also, such as decrease the time it takes to fall to sleep, increase the total time of sleeping, and reduce the number of awakenings during sleep. They have been found to be very useful in promoting sleep as a short-term measure. How do these drugs work on the chemistry of the brain? Studies have revealed that raffinuclei on pons play a very significant role in producing sleep, particularly slow wave sleep. These neurons are serotonergic. It was hypothesized that if a precursor of serotonin is injected into this area, it may reverse insomnia and promote slow wave sleep. To experimentally verify that, the chemical 5-hydroxytryptophan, that is 5-HTP, was injected into raffinuclei of cats and rats. It produced sleep in the experimental subjects as reported in Pinnell 2012. The neurochemistry of non-REM sleep. This category includes stimulants such as cocaine and amphetamines. Some drugs to control depression such as tricyclic antidepressants also come in this category. Such drugs are related with increase in the activity of the catecholamines including epinephrine, norepinephrine and dopamine. The mode of their function is either by increasing their release from the terminal buttons of the presynaptic neuron or by blocking their reuptake by the postsynaptic neurons at the synaptic cleft. Such drugs act on areas involved with REM. They may totally suppress REM. Summary: wakefulness producing and sleep producing substances have been identified. Different brain processing networks and neurochemical systems are involved in both states. The particular neuronal pathways, transmitters and receptors that make up the ascending arousal system centered in the hypothalamus interact with sleep active neurons to produce distinct sleep-wake states with abrupt transitions. The neurons producing acetylcholine and monoamines such as Ne, 5-HT, Da and Ha promote various aspects of wakefulness. In addition, orexins, hypocretins help sustain long periods of wakefulness which suppress REM sleep. Non-REM sleep is mainly regulated by the neural pathways originating in the VLPA and other preoptic regions. REM sleep is driven by neurons in the pons that make acetylcholine and GABA.